Hello, my name is Olivia Stricker, and I'm the Commercial Development Manager at the Diapharma Group. We are located in Westchester, Ohio, and we are a U.S. distributor for many European manufactured biomarker assays, like the ones that I'm going to talk about today. With that, I'd like to start my talk about measuring what matters, exploring two biomarkers for proliferation and cell death, which are TK1 and Keratin 18. So to start off, I thought just talking about generally biomarkers for diseases would be a good place to start. And essentially, reliable biomarkers that are relevant to disease are useful for drug discovery and development, clinical trials, clinical research. But the bigger hope is to be able to take the markers used during those processes and translate them into clinical uses as well. The utilities of an excellent biomarker are shown here, to be specific and sensitive, relevant to disease, quantitative, rapid, robust. For clinical trial purposes, non-invasive sample collection is ideal. And for drug discovery and kind of screening purposes, high throughput is also really valuable. Very few markers can check all of the boxes here. So scientists essentially need to be constantly looking for new tools to use in this process for all disease types. I'm going to introduce two assays that Diapharma represents in the US market. One is TK1 ELISA by a company called Aerosol, and one is an apoptosis ELISA assay by VLV Bio in Sweden. My goal is to tell the story of each of these biomarkers and to present some data from publications that demonstrate their utilities as great research tools in oncology to study new drugs for solid tumors. It's important to note that both of these are for research use only in the US. So keep that in mind as we go through this talk. So I wanted, moving more focused in from just biomarkers for any kind of drug. Good biomarkers are needed for all different types of the cancers that we're studying. By we, I mean scientists. I'm off the bench. But so having good oncology markers on hand is important, obviously. And when you closely look at cancer, it's essentially fundamentally a disease of dysregulation of growth. So when you have cells that won't stop growing or won't die when they're supposed to. Thus, it makes perfect sense that researchers look at markers of both growth and death, and monitoring those two processes, design tests for cancer drugs that tell scientists a lot about the when, where, and how cancer drugs are working during all stages of development. The first portion of the talk I'm going to focus on is cell proliferation. So proliferation is a major deal in cancer because normal tissues don't have a lot of actively dividing cells, while cancer tissues do have actively dividing cells. The special processes that are associated with proliferation have long been targets for cancer drugs. And interfering with these processes are newer ways of targeting these cells for cell death. So used in combo, cancer drugs can basically max out cell damage while simultaneously prevent repair to the DNA. Many of these same drug targets and pieces of the proliferation process might also serve as really great biomarkers for identifying growing cells. And while there are plenty of those available to researchers, and listed here are some of them. Some of them are measuring DNA, some of them are protein measurements. The problem is that not all of these are easy samples to access or easy assays to perform. So some of the most popular protein markers are measured by immunohistochemistry analysis. And while it's useful and effective, it also has a lot of drawbacks in the fact that it requires biopsy, which is invasive, it's labor intensive, There can be sampling issues, which lead to low reproducibility, and there's a certain level of subjectivity involved. So having a blood marker would be with much higher throughput and would translate better to clinical research testing. So one of these markers that's actually currently used for some IHC analysis might be something that would fit that need, and that marker is TK1. So TK1 is a thymidine kinase. It has enzymatic activity, and its key function is in the synthesis of DNA. It's made during S phase and then is degraded after the cells divide. And in cancer events, it seems like TK1 loses this kind of cell cycle associated regulation and is generally upregulated. Now, there are assays for TK1. They're enzymatic in nature, and TK1 is already quite established as a biomarker for blood cancer, as a blood cancer biomarker. But the problem is that many of the assays are radioactive in nature, and thus the activity assay is not ideal. In addition to that, newer data suggests that in the case of TK1, activity levels might not exactly be better than the best measurement to make. And that is demonstrated by the data shown here, where in the case of solid tumors, serum TK1 protein levels have also been shown to be elevated with several tumor types. The example shown here is a prostate cancer type where when the TK1 activity is high, the expression of the protein is low. And when the activity is low, you actually see higher level of protein in the serum. Thus, a critical piece of the information in the amount or level of TK1 protein is being missed in the activity assay. So this drives a need for an antigen-based assay. The current TK1 detection assays out there, again, their activity is still interesting and very relevant to disease. It's a blood cancer biomarker, but the assays are not always easily accessible in the fact that they're radioactive in some cases. In other cases, they're on closed systems or they can be expensive. And this also doesn't fill kind of the gap in the need for detection of antigen for solid tumor purposes. Previous attempts at making a more standard ELISA kit have produced data that show that the readout of the ELISA kit doesn't exactly correlate well with the activity assay that's already been established. The issue with standard ELISA is that TK1 outside of the cell often forms large complexes, and these complexes might be masking the epitopes that are recognized in the antibodies of the ELISA assay. So that being said, a Swedish company called Aerosol has spent some time developing an ELISA kit that addresses the complex problems and really fills this gap in a need for an antigen assay. Aerosol developed an ELISA kit called TK210 ELISA, and they have addressed the complex issues by developing a buffer that dissociates the complexes and increases detection capacity. So when these epitopes aren't masked, you get a more robust assay. And they also have developed a replaced polyclonal antibody with a monoclonal antibody against amino acid 210 of the TK1 protein that increases the sensitivity. So the combination of this results in a more robust ELISA that, as you can see here, demonstrates a strong correlation with the gold standard of the enzymatic activity. So protein correlates with activity. And then also the way that they've addressed it increases sensitivity and specificity so that this kit can be used as a research tool for solid tumors. So shown here is blood donors versus, or serum samples, blood donors versus subjects with solid tumors. And you can see that there is a robustness that might lend itself to being a great tool for doing research on solid tumors. The benefits of the 2K or the TK210 ELISA kit kind of all circle around two advantages that make this kit better suited for cell culture and preclinical work over the activity. Those two benefits or advantages are the fact that the antigenic detection of TK1 is specific and allows for distinguishing between TK1 and TK2, which TK2 is actually found in the mitochondria, but also has thymidine kinase activity. So an activity assay would not be able to distinguish between the two, whereas this antigenic assay does allow for that. That's useful for clinical, like not clinical, but cell culture and other studies on cell-based levels. And then for preclinical assays, TK1 can actually, the TK210 ELISA can distinguish between human TK1 and mouse TK1. And that's, a critical tool when people are looking at human xenograft models and allowing for any TK1 in the blood samples of those animals to be attributed to the actual tumor that's human derived versus the mouse. And, the major advantage with these xenograft models is that you'd be able to measure TK1 in the blood over time, and you don't need to sacrifice animals at each time point since it's in the blood. That translates to clinical research benefits in the fact that they're non-invasive serum-based markers, and this would be advantageous to kind of two different groups of people doing clinical research. So these two groups are kind of who Aerosol wants to get the word out about the 210 ELISA kit. That's the hematology groups that kind of already know TK1 as a biomarker. And for them, the ELISA kit would be more of an old dog with a new trick in the fact that they're used to studying activity, but it might be the case that protein levels are just as good as activity measurements without the hassle of the activity assays. The other group is solid tumor groups that are doing research on solid tumors like breast cancer or prostate cancer. This TK1 in general is a new kid on the block. So the TK210 ELISA kit is a new marker to look at for the research that's already being explored. So that's where the value of this kit lies. And this is an example of some convincing solid tumor data that can be seen when comparing activity as a marker to protein as a marker. So on the top here, we show activity measured in breast cancer subjects, research subjects. And the bottom here is protein being measured in breast cancer research subjects. And the protein levels are much better at detecting subjects with breast cancer and also distinguishing kind of the severity of disease states within those cancer subjects. And in the same publication, the authors also explore a relationship of TK1 protein with breast cancer or with prostate cancer, and they note the same trend can be seen. So keeping in the breast cancer research world, TK1 protein is also a better indicator for subjects with breast cancer than commonly used markers at the present. So in fact, when the markers are used in combination, the data shows an improvement in the sensitivity and specificity. So that would be the black dotted line as compared to the gold standard currently used, which is the blue dotted line shown here. So although the kit is newer to the commercial market, it should be included in all kinds of studies investigating new cancer drugs, especially for preclinical xenograft models, and drug classes that might directly have downstream implications for TK1. Things like CDK4/6 inhibitors might be really interested in TK1 as a biomarker. Here is that extra data for the prostate cancer that kind of also reflect the breast cancer research as a marker, showing that protein is a better indicator in this case than activity for these subjects with prostate cancer. So many more studies are ongoing and publications are much anticipated using this aerosol kit. Aerosol as a company is due to present more data at AACR in the spring of 2020. So if you're interested in TK1, I would stay tuned for that. On to cell death as opposed to proliferation. And cell death would be the flip side of the growth markers, and also to be noted, the desired outcome for cancer cells, all cancer cells. Oh no. So in general, two cell death modalities are kind of more standard or traditional. These two different mechanisms are necrosis and apoptosis. And as I'm sure many people know, necrosis can be thought of more of a homicide cell death due to like infections or toxin or trauma. You would see swelling and loss of membrane integrity, and this kind of results in a lot of inflammation of the surrounding tissue. Whereas on the opposite side of that, apoptosis can be thought of more of a suicide cell death and is very programmed with membrane blabbing and apoptotic bodies that are then gobbled up by phagocytic cells around them and results in much less inflammation. So this is really important when you think about oncology. And while the verdict is still out on whether necrosis or apoptosis is a more desired result for effective cancer therapies, it seems like the evidence is leaning much towards the apoptotic pathway as being preferred. And that's because there's a lot less inflammation in the surrounding tissue. And that inflammation possibly feeds cancer cells. So for that reason, you know, oncologists are interested in apoptosis. Apoptosis has two different kind of starting points. One is intrinsically and one is extrinsically. The intrinsic part of the pathway is activated by a lot of that DNA damaging drugs and checkpoint inhibitors, whereas new classes of drugs are being developed for the extrinsic pathway that actually activate the death receptors and that whole signaling cascade. So apoptosis is a target of a lot of cancer drugs, but it's also a mechanism in which cells become resistant to cancer drugs, and the fact that, you know, they will often bypass a lot of these apoptotic pathways when they become resistant to cancer drugs. So for all of those reasons, and all of those drugs in development, biomarkers that specifically measure apoptosis and necrosis have a tremendous amount of value to researchers focused there. Apoptotic biomarkers that many researchers are using are common and they're useful. They're listed, some of them are circled in red here, like cytochrome C and nucleated DNA, changes in the membrane, activated caspases, and cytokeratins. The desired qualities would be much like any good biomarker in the fact that they're apoptosis specific, quantitative, they're high throughput for drug screening, that they're an endpoint marker, that you don't have to capture a cell in the middle of apoptosis in order to get this readout, and that they can be found in the blood. So unfortunately, many of the desired qualities aren't delivered by every single biomarker. You know, they might not be specific to apoptosis, or the product of apoptosis might not be stable for effective quantitation. So I thought I would explore the cytokeratin as a biomarker more in depth. The cytokeratin, our favorite cytokeratin here at Diapharma, is keratin 18. So it's obviously one of many different keratin types. And just like any good protein, it has many names depending on the area of research that you're in. It can go by keratin 18, it can go by cytokeratin 18, K18 or CK18. It is found in mainly epithelial cells of the body and their tissues. So liver, lung, intestines, breast, prostate are some main ones. It is not expressed in the brain or neurons, muscle, connective tissue, and importantly, immune cells or cells of the blood. And that's attractive for the reason that any massive cell death in the immune system actually won't interfere with measurements of keratin 18. So that's an advantage, especially in oncology. And also there's a laundry list of publications that kind of associate increases in serum levels of keratin 18 with the presence of tumors in things like the liver, the lung, the intestines, or breast or prostate cancer. So there's a lot of interest to oncology land. If you look at cell death in the context of keratin 18, in necrotic conditions, you know, total intact keratin 18 can spill out into the surrounding cell. Whereas under apoptotic conditions, you get activated caspases that will cleave keratin 18 at very specific amino acid sequences. So the result in an apoptotic cell is that you get lots of small pieces or fragments of keratin 18, either packaged into these phagocytic bodies or spilling out into the extracellular space around the cells. Diving in to look really specifically at keratin 18, as I said, when apoptosis occurs, it actually gets cleaved at two specific locations. And keratin 18 has proved to be of significant interest primarily because of several monoclonal antibodies that have been developed against the protein. So they've been developed and then they've been characterized by the Lindner lab at Karolinska Institute, and then commercially developed further into ELISA kits by a company called Paviva, which was then purchased by another Swedish company and is now known as VLB Bio. These antibodies are called M5, M6, and M30, and the M5 and M6 recognize the body of the protein, keratin 18 protein, whereas the M30 is a very special protein, or very special antibody in the fact that it specifically recognizes the caspase cleaved keratin 18. So it only sees the neoepitope that was created after that cleavage occurs. And it also goes by a barrage of names. Some of them are names of the neoepitope, then some of them are caspase cleaved cytokeratin 18 and caspase cleaved keratin 18. So you'll see the apoptotic product referred to as all of those names. It's also important to note that the M30 antibody name has been trademarked, and in addition to the ELISA kits that have been developed, they are also trademarked names. So M65 and the M30 ELISA, M65 is comprised of the M6 and the M5 monoclonal antibodies. They detect both the full-length intact keratin 18 as well as the apoptosis product, whereas the M30 antibody only recognizes that neoepitope. So the ELISA kit that contains the M30 antibody only recognizes apoptotic products. Used together, these two products are very powerful in the fact that the ratios of the two can tell us about cell death mechanism, which in the land of drug development is actually very important, whether things are necrotic or apoptotic. So when you have high apoptosis product measured by M30 with high total keratin 18 measured by M65, that indicates an apoptotic event. So here I show data with a breast cancer cell line treated with a drug that induces apoptosis called cisplatin. So when you add cisplatin, you see that you get an increase in both the apoptosis and the total keratin 18 product. And that the product, to note, is actually stable over time. You see an increase from 24 hours to 48 hours. And if you treat those cells with an apoptosis inhibitor, the ZFAD indicated here, you can actually block the apoptosis product from accumulating, but not the total keratin 18 from accumulating. So that would be an example of what an apoptosis event looks like. On the other side, a necrosis event, the same cell line treated with necrosis-inducing drug called oligomycin. Induces total keratin 18 in the media, but does not produce high levels of the apoptosis product in the media. So while that is a characteristic that is very powerful for a lot of cell culture and screening work, other characteristics of the M30 antibody is that they're specific. It is specific for keratin 18 and apoptosis. So shown here in this figure, I have, not I have, but the author, I didn't do any of this work. The author shows that CK18 is only detected when it's transfected into these CK18 minus cells, and that the neoepitope is actually only detected when those cells are also treated with an apoptosis-inducing drug. For cross-reactivity studies, data shows that mouse, rat, or dog peptides don't compete off the human peptide when binding to M30. So therefore, we can say that M30 does not detect mouse or rat apoptosis keratin 18 product. That is extra important for any xenograft models that are done. So just like the aerosol kit, the M30 Apoptosis only recognizes the human caspase-cleaved keratin 18 in the serum of these animals. And there are papers demonstrating that you can use mouse or rat models and a variety of tumor models and a variety of drug types to study the pharmacokinetics and the pharmacodynamics of how drugs act on different tumor types. So this is an example of data that helps demonstrate that, where in this first part of the figure, they're using a mouse model with a head and neck tumor, and you can see that over time, you actually get a very dramatic increase between untreated and treated tumors in the amount of apoptotic product that happens. So look, if you look at 0 and 96 hours, there's a lot higher levels of apoptosis going on. And then if you take that same model, you can do a dose response curve as well and get really nice tight dosing information. This same author replicated that work in a rat model of colon cancer and showed that, the assay is reproducible in a plasma assay. So that's really powerful use in xenograft models. Lastly, I'm showing you data that, see, on the left, a prostate tumor is not responding to a cancer drug, indicated by the increase in the PSA levels. And the CK18, the apoptotic product, remains flatlined. On the right, when the cancer drug is effective, the tumor shrinks and the PSA level plummets. And it's observed that the CK18 actually peaks with each cycle given to the subject between day 5 and day 7. So this data suggests that serum CK18 measurements may be useful for assessing the treatment effects. And the method, is kind of ideal for clinical trials in the fact that the product is stable in the serum and can be stored for a long period of time and is ideal for kind of multi-center clinical research trials. The measurement of the caspase-cleaved keratin 18 in the serum might be useful for comparisons of efficiencies between different drug modalities, and that's a pretty key issue during development of novel anti-cancer drugs. So there's a lot more data out there to look up. I just don't have time to go through all of that today. But I appreciate everyone for bearing with me and allowing me to give you the take-home messages that there's 2 biomarkers that should be included when looking at especially xenograft models. One is TK1. At the protein level, there's a new kit by Aerosol. It's called TK210 ELISA, and it is more sensitive and robust than any kit that's been available in the past and better than the activity measurement for solid tumor. research. And the other marker is cytokeratin 18, specifically the apoptotic product that's measured by the M30 Apoptosis ELISA kit. It can be used in tandem with the M65 ELISA kit to kind of give a lot of extra information on the mechanism of cell death, and can provide kinetics in human xenograft models to do solid tumor studies as well. So both of these can be used through the whole drug development process. And with that, I just wanted to remind everybody that that Diapharma is the company that offers these products in the US. We are the exclusive distributor for VLV Bio. We kind of specialize in a lot of really technical, technical support, and can provide a lot of input or extra data, or answer any questions about these specific products. We have these as well as many other oncology markers that are kind of more unique in nature, and they can all be found at our website, which is diapharma.com, or you can feel free to reach out to any of our team members at the contact that I have listed here. Also, we will be at AACR in the spring of 2020, and you can always find out what our latest and greatest is on our website or on our LinkedIn. So with that, I'm finished. Thank you.